Okay, chapter 4 is titled Cell Structure, and I'm not going to cover the entire chapter because it's huge. And so for today, I want to talk about, um, well, a cell theory, and then talk about prokaryotic cells and how they compare to eukaryotic cells. And that's all I'm going to focus on today. Tomorrow, we'll talk about the endomembrane system, as well as all those organelles inside the cell. So we have quite a few objectives in chapter 4, but I'm going to cover the understand why most cells are small in size, and there's at least 4 factors there. I'm also going to describe the function and composition of the plasma membrane, the principles of the cell membrane, and then differentiate between prokaryotes and eukaryotes, and then finally talk a little bit about the structures of the nucleus and its components. So starting off with the cell theory. So cells are really, really small, microscopic size. But there is a difference between eukaryotic and prokaryotic. Prokaryotic cells tend to be a lot smaller, and the reason for that is because they don't have all those extra organelles that eukaryotic cells do. Prokaryotic cells don't even have a nucleus. So I always tell my kids, I know pro usually means like you're for something, but in this case, we're just going to say pro rhymes with no nucleus, okay? Eukaryotic cells do have a nucleus. Eu means true, and then kary means kernel or nut, so it's a true kernel or true nut. Um, and it's describing the nucleus that's in the center of the cell. So they have a nucleus. They also have a lot of organelles. I want to go back to prokaryotic cells later on today. Um, I shouldn't say they don't have any organelles. They do have an organelle, and it's the ribosome to make proteins, but they don't have like an endoplasmic reticulum or a mitochondria. So, but the discovery of cells obviously didn't occur until we could see them, and so the invention of the microscope really helped us advance, what is life really made up of. And so we start with the story of Robert Hooke in the 17th century, where he looked at dead cork cells, and he noticed that there were these tiny little openings, and he's like, oh, they look like cells. So he's actually the person responsible for the coined term for cells. Anton von Leeuwenhoek um, looked at pond water and noticed that there were tiny little organisms swimming around in pond water. He actually called them animalcules, but he's kind of credited for looking at the first live cell underneath a microscope. But the cell theory took over 150 years to develop, and it's actually, many scientists, many observations put together over a series of time. Remember, theory and science is not a hunch. Theory and science is absolute, it's fact. Okay, it's based on evidence, and until we come across evidence that doesn't fit, then we have to go back and revise the cell theory. But this is based on observations and experimentation. So when the microscope came out, everyone wanted one. Everyone started to make one, and they started to look at everything underneath the cell. And so Matthias Schleiden, was looking at plant tissues underneath the microscope, and every plant that he looked at underneath the microscope were made of these cells. Okay, Theodor Schwann started looking at animal tissues. Cow, dog, sperm. I mean, don't ask me where he got the sample from, but made this conclusion that all animals are made up of cells. Okay, and then the third principle is put together by a guy named Virchow. And the third statement is that all cells come from cells. Cells come from cells. And he actually noticed cell division underneath the microscope, a split of one cell becoming two cell. And everyone thought he was crazy when he made this proposal, but then other scientists also made the same observation. And so, Virchow's kind of credited for that third statement. All cells come from other cells. So here's the 3 principles of the cell theory. All organisms are composed of one or more cells, and the life processes of metabolism and heredity occur within these cells. Usually I just say all organisms are made of cells. Cells are the smallest living things, the basic unit of organization of all organisms, or maybe you learned in biology or life science, cells are the basic unit of life, okay? Until we come across a living thing that is not made up of cells, this statement will hold. And then cells arise only by division of a previously existent cell. So cells come from other cells. Okay. Now I know we've talked a little bit about um, our hypotheses, where we've made the jump from chemical life to biological life. But, you know, this is where we're at right now. Okay, cell size is limited. Like I said, cells are microscopic, and there's a reason for that. A huge part of it has to do with its diffusion rate. So cells have to be able to move things in and out of its membrane from one part of the cell to the other. And so the rate of diffusion affects the surface area. So as you um, increase your surface area, the oh my gosh, why can't I think? The volume increases, and then the rate of diffusion increases, and that's bad for a cell. If it can't move things as fast as it needs to, it's going to struggle, it's going to die, it's not going to function properly. Temperature also affects the rate of diffusion as well as concentration gradient, and we'll talk a little bit more about that in Chapter 5, and you guys will actually have to do kind of a lab write-up on that. It's a new thing with MSTAY, so I'll explain more when we get to Chapter 5. As cell size increases, the length of diffusion increases as well. Large cells need to synthesize more macromolecules, so they need more energy. And then obviously they also have to be able to remove waste, because if they can't get rid of it, then that waste becomes toxic to them. And then again, things are not going to work right, and it could possibly die. Now in your book, it says something like list 4 factors or variables that may affect the rate of diffusion. In your book, they have it numbered really, really nicely. So I encourage you to take a look at your book to answer that question. Surface area to volume ratio. Okay, so here, as the cell increases, volume increases more rapidly than the surface area. So here's my volume of 1 meter squared, millimeter squared, sorry, wow. Surface area 6 millimeters squared. And then as you increase just by 1, now you have a factor of 3 versus 6. And so the higher surface area to volume ratio, the better for the cell. Okay, going back to microscopes a little bit. So microscopes allowed us to see cells. However, there is a problem with microscopes, and it's called the resolution problem. So sometimes we can't see cells because of our resolution. So definition of resolution is the minimum distance 2 points can be apart and still be distinguished as 2 separate points. So I just love this diagram. Resolution allows us to see objects as separate from one another. So with our physics going on with our eyes, as well as lenses with microscopes, we can't see everything with just a typical light microscope, but we can see a lot. We can see hair, we can see an ant, we can see cells. Bacteria, you probably need a more high-powered microscope, but we won't be able to see that with our microscopes. And then with the super resolution, viruses really, really small because they infect bacteria, proteins. Can't really see molecules. There are 2 types of microscopes. A light microscope means that it uses visible light and 2 magnifying lenses, and the compound just means that there's lots of stages, and you can change lenses several ways. So one way to increase our resolution is to increase magnification. So then you're probably thinking, well, geez, can't we just keep adding magnifying stage to a microscope to really magnify things? Well, you can't, because there goes the physics. Light beams from 2 objects overlap with each other, so we have to have a way lower wavelength in order to do that. And to achieve that, you actually have to think about the electromagnetic spectrum and jump below the visible light. So we have electron microscopes. 1,000 times the resolve and power than light microscope. There are 2 types. Don't get them mixed up. Transmission means that it literally goes through the specimen. Okay, so here is a cross section. The electrons are going through the whatever sample that you're looking at. Okay, scan on electron microscopes give these awesome images, these 3D images. So these are the same thing. Okay, cilia, but here we're looking at the outside, and here we're literally going through the organism. Okay, This is a light microscope of a cheek cell. So you'll actually get to check out your cheek cells again this year, I think, when we get to, I want to say, mitosis. Okay, we can use stains to help us view cell structure. There's lots of different stains that can bind to different proteins. There are stains that bind to DNA, so that's DAPI blue, and DAPI blue is always used in DNA. We have red fluorescence and green fluorescence. A lot of times, like, this is like one part of the cytoskeleton, and this is another part of the cytoskeleton. I know we haven't talked about the structure of the cell yet, but the cytoskeleton is made up of 3 different fibers, and so each of these will bind to a different fiber. And when you overlap all 3 of these colors, blue, green, red, you get images like this. Okay, and it's telling a story here. Sometimes you can stain molecules. So when I was doing my research with circadian rhythms, with that whole, trying to mutate cry, make it bind to PP5, we actually stained cry and PP5, and one was we used like a light blue, another one was red, and so we were looking for a deep purple color, and it never, like, it left the nucleus. Like, it wasn't supposed to leave the nucleus, but all of our samples showed that it left the nucleus, which means that it was interacting and doing its job. So our mutations, we didn't mutate it where we wanted it to. So, but colors, they can help you trace and follow molecules. Okay, all cells do exhibit some basic structural similarities, however. So they have genetic material, and it's usually centrally located. However, I do want to point out prokaryotes don't have a nucleus, okay, but it's in the center of it, or of the cell, and we call it the nucleoid, okay. Another thing about prokaryotes and their DNA is that it's a circular molecule, okay. In eukaryotes, we have it in the nucleus. It's a double-bound membrane, and in most eukaryotes, it's linear. I should have put that in there. So that's one similarity, genetic material. Number 2, they have a cytoplasm, okay? Cytoplasm fills the interior of the cell, contains sugars, amino acids, proteins, just to carry out all the chemical reactions. I mean, a lot of chemical reactions take place in the cytoplasm, and you don't even know it. It's not water, it's more like jello. Contains your organelles, and then parts of the cytoplasm can be called the cytosol, which means if it has organic molecules and ions in solution, we are going to call that the cytosol area. Another characteristic that all cells have is they have a plasma membrane. So it keeps contents in and out, so it's a boundary. It's made of a phospholipid bilayer, and there's proteins embedded into it. And those proteins do interact with other molecules as well as the environment. So we have proteins that help transport things, and we also have proteins that receive chemical messages, and this can also serve as an identification marker for a cell. So you have cells in your bloodstream that have these protein markers to say, yep, I'm a part of you, and you have white blood cells that every once in a while will just do like an ID check. Yep, we're on the same side. But if it ever comes across a foreign particle, and it's going to be like, okay, you don't belong here, sends off another signal transduction pathway, recruiting more white blood cells, macrophages, to get rid of that foreign particle. So it's like, this is very important. Autoimmune diseases, this is one of the reasons, that is the reason why people have autoimmune diseases, is because their identification marker on their cells, they don't recognize that they're on the same side, and so their cells literally attack their own cells. The 4th characteristic that all cells have is that they have ribosomes that make proteins. So I know when I say prokaryotes don't have organelles, okay, they do, they have a ribosome, but other than that, they don't have all the other organelles. Okay, so prokaryotic cells don't have a nucleus, and they don't have this internal membrane system or organelles except for those ribosomes. So if we take a look at kind of a flow chart of cells, here we have cells. All living things are made up of cells. You can be prokaryotic or eukaryotic. And if you are prokaryotic, you can be part of the domain eubacteria or bacteria. or archaea. Um, and so we're going to talk about these two in this section. And then in 4.3, we'll talk about eukaryotic cells. But I'm not going to talk really about plants, animals, fungi, protista. That's more like semester 2 stuff. But we will talk like, oh, there's a difference between plant cells and animal cells, and especially when we get to mitosis. But prokaryotic cells do have this kind of simple organization. I mean, really, they are the simplest organisms. They are unicellular. They're small. They have a cytoplasm. Um, they have a plasma membrane that keeps that cytoplasm inside. They do have a cell wall, and so I'm going to talk a lot about the structure of the cell wall. No organelles except for those ribosomes. They don't have an elaborate cytoskeleton because they don't need to. They have a cell wall, and that's what gives them strength. Two domains of prokaryotics, archaea and bacteria, and they are found in all ecology of life. They are responsible for diseases. They can be living things. The cyanobacteria billions of years ago are responsible for the oxygen we breathe today. So photosynthesis, that's what the PSN is abbreviated for. I'd also like to say decomposers. If we didn't have bacteria that could break things down, could you imagine a world where things did not decompose? So like a tree falls in a forest and it just stays there and it never breaks down. Or, it would blow my mind if things never decompose. Things would just pile up. Okay, so let's talk a little bit about that cell wall. So their cell wall is different from plant cells. Plant cells, their cell wall is made up of, this should be review. Hopefully you're thinking cellulose, but that's okay if you're not. But the cell walls are made up of peptoglycan here in bacteria cells. And so what is peptoglycan? It's a carbohydrate matrix. Lots of crosslinks going on here. Very, very strong. Helps maintain shape of the cell. Helps it from taking in too much or losing too much. So it maintains its shape. Plants, fungi, and most protists have cell walls, but they don't have peptoglycan. So peptoglycan cell walls is what kind of distinguishes a bacteria cell. Now, there are two types of cell walls, okay? You can be a gram-positive or a gram-negative, and a lot of it has to do with stain, okay? So in a gram-positive, you come across a peptoglycan cell wall that's thick. It is a single layer peptoglycan, and it retains a violet dye from a type of stain, which we call a gram stain. So they're purple underneath a microscope. Gram-negative are way more complex. Multi-layered cell does not retain the violet dye, so they end up pink or red underneath the slide. And so gram-positive and gram-negative cells actually tie into antibiotics. Some antibiotics work on, gram-positive cells. What they do is they drill holes into that single layer peptoglycan cell wall and they flood the cell. That's one way antibiotics can work. Other ways is they somehow get into the bacteria cell and they mess up the ribosomes, because if you can't make proteins, if you can't make stuff that the cell needs, it's gonna die. So antibiotics, there's different ways that antibiotics work. Gram-negative cells, a little bit more difficult to fight, and the reason for that is they tend to secrete some kind of jelly-like capsule that allow it to just kind of be slick and slippery. Okay, so here's that gram-positive single-layer peptoglycan cell wall outside the cell membrane, and you can see that it's got teichoic acid and lipoteichoic acid. So they're just like kind of proteins, receptors on the outside of the cell that can communicate with inside. The gram-negative, multi-layer. Okay, so notice on the outside we have these green, heads, if you will, lipopolysaccharides. Okay, so we actually have like a fat carb matrix going on here. There's the peptoglycan, a lot thinner, and then there's our plasma membrane. And so you can see that it secretes some kind of slippery jelly mucus-like, and it's the lipopolysaccharides that make it gram-negative cells a little bit more difficult to fight. So here's the stain underneath. So the staining process is that, they take the sample that they want to test, and they apply a crystal violet, it's a purple dye. Then they add some iodine to it. You guys messed with some iodine in the Chemicals of Life lab. Then they wash it with an alcohol, and then they add saffron as a counter stain, and then what you see is what you get. So purple is gram positive, and negative is pink, or yeah, pink. So don't get those two colors mixed up for what they mean. You need to know, pink is gram negative and purple is gram positive. Now, archaea lack a peptoglycan wall. We're still learning a lot about these organisms. The reason for that is they're really hard to culture in labs. You actually have to go out into the extreme environments that they are found. They are found at the very bottom of the ocean floors. Yellowstone National Park, very like the hot baths, the pristine pools of Yellowstone, just like extreme environments, even in the cold Antarctica. So like these guys are really interesting to study because like when we look for life elsewhere, out in the solar system, the life that we may encounter if we ever do was probably going to be a type of organism like this that can withstand extreme conditions. So their common feature that distinguishes them from the bacteria is they just have different lipids. And the lipids are saturated hydrocarbons. So remember, saturated means that they have single bonds, no double bonds. And they are covalently attached to glycerol at both ends. So we actually have a mono layer here. And this works for them because now they have greater thermal stability, where they can withstand extremely high temperatures. But it kind of makes them lose this ability to adapt if their environment should ever change. So here's a Venn diagram kind of comparing the two bacteria archaea. So cell walls of peptoglycan cannot survive in extreme environments, but they are found in all environments. Over here, live in extreme environments, cell walls made up of different matter and less standard energy production. They're not really responsible for photosynthesis. But how they're similar: no nucleus, no organelles, single-celled prokaryotes, probably evolved from a common ancestor. Now, like the slide, this is actually showing the plasma membrane. And you can see that we have one phospholipid here with the two fatty acid tails, and another phospholipid head with the two fatty acid tails, and they are not connected, okay? These tails interact. Sometimes they are kinked, okay? In archaea, notice that they are covalently linked, okay? So they're monolayer. That's what they mean by monolayer. However, there are some species that do have a bilayer, okay? But monolayer, probably the most distinguishing factor between them and bacteria. Okay, DNA and synthesized proteins, however, are more closely related to eukaryotes in archaea than they are to bacteria. So at first, people thought that, okay, they're one cell, there's no nucleus. Yeah, these guys branched away from each other. Like that's where they thought archaea should have been, but that's not the case. When you look at their DNA and the proteins that are made, they actually resemble more closely to eukarya, okay, organisms that have a nucleus. And so now we think that it was bacteria and then eukarya, and then from there maybe it branched into eukarya or archaea, or maybe it's vice versa. Cells branched into bacteria, archaea, and then eukarya derived from archaea. I don't know. Okay, so here's the cell types. Nice little table here that does a great job comparing all three of these domains. I shouldn't say domain, well, yeah. So eukaryotic. We haven't talked about histones yet, but they do have histones. Okay. When you look at archaea, have proteins similar to histones. Introns, we haven't talked about introns yet, or the ribosomes. The cell walls. We know that there are some eukarya that have cell walls, and plants are cellulose, fungi, chitin, and there's some membrane composition. Eubacteria, that's bacteria, so no histones, no introns. They do have the same ribosomes, cell wall made of peptoglycan. We haven't really talked about external-linked lipids yet, but we'll get there. Okay, some prokaryotes can move by rotating flagella, And so flagella is this propellant-like tail that's used in locomotion. They can swim extremely fast with a flagella, 70 cell lengths per second. There are different types of, well, I should say, you can have lots of different types of flagella, but um, one-tailed, two-tailed, multi-tailed, tails everywhere. Um, and so we're going to come back to flagella because eukaryotic cells, some of them can also have flagella, but the structure of their flagella is a little bit different. So that's another difference that prokaryotics have with eukaryotics. Now, eukaryotic cells are way more complex. And the reason for that is they have an endomembrane system, which means we have lots of membranous compartments that carry out specific functions for the cell. We have organelles. Plants have that central vacuole, and so the central vacuole in life science and biology, we learned that it stores water, but it can also store pigments, proteins, as well as waste materials. Both have residuals, smaller sacs that store and transport material. We're going to talk a little bit about the nucleus. Actually, we're going to talk a lot about the nucleus in this section. And then all eukaryotic cells have a cytoskeleton, and so I think we'll dive into the 3 parts of the cytoskeleton too. But here's an endomembrane system, a visual summary of where we're going to go. We're going to start in the nucleus and work our way to the endoplasmic reticulum, where it can be go through the smooth endoplasmic reticulum or the rough as vesicles to the Golgi apparatus, and then it can either leave the cell or meet up with a lysosome or whatever. But it's all these tiny little membranes that interact with each other. But we're going to start with the nucleus. We all know that it's the information center. It stores genetic information. It's the largest, easily seen organelle. When you look at your cheek cells underneath the microscope, it's the only organelle that you can really see. But Robert Brown, 1831, kernel or nut, kind of coined the term nucleus. And so there are different parts of the nucleus we have to be aware of. The nucleolus is the dark staining zone. Lots of intensive synthesis of rRNA is taking place here. So there's the nucleolus. Then we have chromatin, which is DNA plus proteins. Those proteins are called histones. And then we are going to talk a little bit about the nuclear envelope, how it's also double membraned, how things leave the nucleus through the nuclear pore, and then where do they go after that? Usually the ER. So the nuclear envelope is bounded by 2 phospholipid bilayers, okay? And the outer membrane is actually continuous with the ER, the endoplasmic reticulum. This is actually kind of cool because during mitosis, you have prophase, okay, where the chromosomes condense. And then you have metaphase. So in prophase, the cell membrane starts to break down, and then in metaphase, there's no nucleus. It's like, where did the nucleus go? Well, it actually gets absorbed by the endoplasmic reticulum. And then when the cell is done going through cell division, then the ER, helps reform the nucleus. So it's kind of a cool thing. The nucleus has several nuclear pores. It's very complex. It has a cytoplasmic face, which means it faces the cytoplasm, has a nuclear face, the part that faces the inside of the nucleus, and then a central ring that we'll talk a little bit about. But it allows small molecules to diffuse freely between the nucleoplasm, so the stuff inside the nucleus, and the cytoplasm. Passage is restricted primarily by 2 kinds of molecules, proteins and then rRNA and their protein complexes. So proteins that are catalyzed near the nucleus can enter or leave, and then we have rRNA that's usually exported to the cytoplasm. So here's the structure of the nuclear pore, and I just love these diagrams here. So here we have double membrane, okay? And we are facing the outside of the nucleus, okay? So this would be the cytoplasm face, and then this would be the nucleoplasm, and you can see that there's chromatin, that's proteins with DNA, and it kind of looks like kind of a basketball net. So here's a ring, and I don't know what you want to call these things, but it's like a double ring structure, and then we have these crazy looking little tails on the outside that help latch on the molecules and literally dunk them in the basketball hoop. So another diagram just kind of showing this ring subunit, double ring, tails. Here's transmission electron microscope. Okay, and these are scanning electron microscope images. And so these are what the nuclear pores literally look like outside of a nucleus. And you can see that there are numerous. Okay, um, so fibers are intermediate filaments called the nuclear lamina. Gives the nucleus its shape. Let's see if I can find the lamina in this. There's the lamina right there in yellow. Involved in deconstruction and reconstruction of the nuclear envelope during cell division. Now, I do have a trippy video to show you. It's trippy. Okay, so I hope it works here. Yes. So there's the nuclear pore complex. Cytoplasmic filaments, just, I don't know, tentacles. So here's a protein molecule that needs to get inside. Got latched on by a tentacle. Now you are the protein going through the nuclear pore. So violent. And then it gets into the nuclear basket. Go. There we go. Yep. Short, trippy little video. And there is no sound, so. Okay, chromatin. So inside the nucleus, we have chromatin, which is DNA, but it's DNA kind of wrapped around proteins called histones. So if we take a look at our double helix, DNA double helix with our nucleotides, and then we wrap around beads of histone to help supercondense, and then these wrap around each other, and then they coil and coil even more and become even more supercondensed, and there's our chromosome. Remember, DNA is huge. I'm sure if you remember last year, the short little video I showed, if you were to take your DNA and just uncoil it, okay, from one cell, the cheek cell, it would be 9 feet long. How in the world do cells fit all that inside of a microscopic cell? The answer is all of this coiling and supercoiling, to get it to fit. And then not to mention that you have 46 chromosomes, and there are some cells, some organisms that have way more, like carp has 104, okay? So anyways, prokaryotes, their DNA is organized into a single circular chromosome. Eukaryotes, multiple linear chromosomes. So chromosomes organized with proteins is when we call it chromatin. So the proteins are called histones, and when DNA is wrapped around histones, nucleosomes are formed. So just to give you a little recap here, chromatin is DNA plus proteins, and those proteins are called histones. And when DNA is wrapped around histones, now they're called nucleosomes. So this is a nucleosome. DNA does have the ability to loosen or extend so that proteins can interact and attach, as well as to compact into highly condensed forms during cell division so it doesn't lose bits and pieces of it as it's moving into the new daughter cell. Okay, so DNA double helix wrapped around histones. Now you get nucleosomes, supercoil along each other to chromatin. Okay, little comic strip. Hope you finished condensing your DNA. We're dividing in 10 minutes. Aww. All right, the nucleolus, ribosomal subunit manufacturing. So ribosomes make proteins, and a cell obviously needs a lot of proteins because everything's made up of proteins, and proteins carry out chemical reactions. So there's hundreds of copies of genes that code for our RNA, and they're clustered throughout the chromosome. So it's this area of the nucleolus is responsible for making ribosomes. The nucleoli, ribosomal assembly areas within the nucleus, and it's that dark stained area that you can see with a light microscope. Now, ribosomes, main protein synthesis machinery in cells, and ribosomes made up of 2 rRNA molecules, a large subunit and a small subunit, And when they attach and form, then you get an actively synthesizing protein. Making protein requires two other forms of RNA, and we'll talk more about that in transcription and translation. But messenger RNA and transfer RNA. If you remember last year, messenger RNA carries the code and information from DNA. It's the thing that actually leaves the nucleus and finds the ribosome outside in the cytoplasm, or the rough ER. And the transfer RNA was the broken dynamite stick figure that carried amino acids to the ribosomes, and then the ribosome helped form that peptide bond between the amino acids to make proteins. My last slide. Ribosomes are free in the cytoplasm, or they are associated with internal membranes. And so you can find free ribosomes in the cytoplasm. Nuclear proteins, mitochondrial proteins, mitochondria actually have their own type of ribosomes. Or you can find them on the rough ER. But anyways, ribosomes are universal machines, and they are found in all three domains, even though the structure of it could be slightly different. So that's all I wanted to talk about today. Tomorrow we'll talk more about the organelles, the endomembrane system of eukaryotic cells. So